The purpose of this video is to give you a little bit of an introduction to the enzyme lab that we'll be doing in class. It's going to be a very busy lab. We'll have lots of things to do, and I'm hoping that we can get it all done. So the reason I'm giving you this is so that you can prepare for that, and we don't have to do this during class, during the lab period. So just by way of brief introduction and reminding you, we are going to be dealing with amylase, which is an enzyme that breaks down glycosidic bonds in starches in order to form simple sugars. Okay, and the primary sugar that's formed is actually maltose, which is a disaccharide. So you can see here that starch is broken down. The alpha glycosidic bonds are broken to form maltose. And we have amylase in our mouths. We start to break down food starting in our mouths. And then we also have a lot of amylase that gets dumped into the system by our pancreas. And it continues to break down starches at the far end of the stomach. So recall previous labs where we've learned how to test for different types of molecules. How do we test for, we can test for both reducing sugars like maltose, and we can test for starch, okay? The test for reducing sugars is the Benedict's test, and the nice thing about the Benedict's test is that it's negative at the start of the reaction, and then as soon as we get some reaction happening, we should get a positive result, indicating that some appreciable amount of maltose is present. Okay, the test for starch is a little bit different because instead of testing the end product, we're testing the presence of the reactant, the starch. And so we expect to get a positive result at the beginning of a reaction, and we expect to get that as the reaction progresses, we still expect to get a positive reaction until the reaction is complete. When the reaction is essentially complete, there's no starch left, then we should get a negative result with the iodine test. And it's this iodine test that we're actually going to be using for this lab. So the objectives of the lab are to first investigate conditions which affect the speed of reaction. Okay, and the main condition we're going to be looking at here is our pH, and we're also going to look at how the substrate concentration affects the speed of reaction. And when we do that, we're actually going to estimate how fast the reaction could possibly go, how fast the enzyme works, using something called Michaelis-Menten dynamics. So let's just review just a little bit. The binding site of an enzyme the active site of an enzyme is where the enzyme binds to the substrate. And this binding usually happens primarily through hydrogen bonds. And because of this dependence on hydrogen bonds, the pH of the solution really matters a lot. An optimal pH is going to be different for each enzyme. Some enzymes will work happily, like the pepsin in your stomach, at a very low pH, a very acidic environment, and some enzymes will need to work at a much higher pH. But for each enzyme, there's an optimal pH that it works. And if you go too far, if you go to pHs below or above that optimal, then the speed of the reaction drops off. We have lower enzyme activities at those extreme pHs. Okay, we also expect the concentration to affect enzyme activity. If you think about chemistry, you probably know that the more concentrated your reactants are, the more quickly the reaction proceeds. That's just simply because there are more collisions happening in the beaker or whatever. So let's think about it in terms of the enzyme itself. If these dots here in the beaker are the substrate, so they're not the enzyme, they're the substrate, but we add a certain amount of enzyme to these to these beakers, the same amount of enzyme, in which beaker would the reaction happen the fastest? Okay, you should recognize that it would happen fastest in the concentrated solution, even though the number of molecules of enzyme is exactly the same. And the reason for this is that the enzyme spends a certain practically fixed amount of time actually carrying out the reaction. So once the enzyme encounters the substrate, it takes some amount of time for it to carry out that reaction. Usually it happens very, very quickly. In the dilute solution, however, the enzyme basically spends most of its time almost like looking for substrate molecules. It's just kind of bouncing around, waiting for substrate molecules to collide with it. And so the reaction rate is relatively slow. Most of its time is used looking for or waiting for a collision. In a concentrated solution, however, the enzyme still spends a fixed amount of time on every single reaction that it carries out. But as soon as it's finished with one reaction, another substrate molecule collides with it, and it can start working on that next substrate molecule. So in all, it spends very little time looking for substrate molecules to react on, and so the reaction rate is generally quite fast. If we plot this on a graph, we should get a graph that looks like this. Okay, so on the left side of the graph, you have a dilute solution. On the right, you have a concentrated solution, and the y-axis here is the reaction velocity, or how fast the reaction happens. Now let's just look at the very bottom left corner here. At these concentrations, the substrate is very rare in the beaker, and so the reaction happens very, very slowly. Very few molecules are actually converted. If you go over to the top right here, to the right side of the graph, then the substrate is very concentrated, and molecules are bumping into the enzyme all the time, and the enzyme spends very little time looking for a substrate, and so the reaction rate is very quick. Now, if you were to extend this graph way further off to the right, then you would eventually get to the maximum velocity of the reaction, the Vmax. And this is the rate at which the enzyme basically spends absolutely no time, practically no time, looking for substrate molecules. Whenever some products leave, new reactants immediately come into the active site because they're so concentrated. So there is a maximum rate at which the reaction happens, and this is the speed. Basically, this indicates how fast the enzyme can actually carry out the reaction with no sort of searching time. Okay, so for this lab, the lab comes in basically three parts. The first part, we're going to estimate how much amylase to use. Okay, so the amylase again is the enzyme, and we're going to be operating it on starch. So how much amylase can we use to get a pretty quick reaction? So we can't wait all day for this reaction to happen, but we don't want the reaction to happen instantaneously because it's really hard to measure. You'll see in the lab, but it's really hard to measure something that happens that quickly. So we want to estimate the amount of amylase and starch, the ratio of amylase to starch, to be so that the reaction takes place over a couple of minutes. Okay, and in order to do that, we'll use a set amount of starch, and then we'll vary the amount of amylase. So to do this, we're going to make a serial dilution. Okay, and the serial dilution is basically a way of making a dilution where you take the dilution present in one tube, and you make the next tube, you dilute it into the next tube, and then you take that solution and dilute it into the next tube. Okay, so to do this, we're going to first of all start by adding buffer to each tube. Okay, then we're going to add amylase to tube 1 and mix it up. Okay, so tube 1 will contain a mixture of buffer and amylase. Then we're going to take some of that amylase, okay, half of that amylase, half of that mixture from tube 1, and we're going to add it to the buffer that's already in tube 2. Then we're going to mix those and take that mixture from tube 2 and add it to the buffer that is already in tube number 3. Okay, and so you can see as we go down the line, we're going to get more and more and more dilute enzyme. Okay, and actually I've listed the dilutions here. This graphic only goes to 7 tubes. We're actually going to go to 10 tubes and include a control that does not have any amylase at all. Okay, so we're going to have this range of tubes, and then we're going to see how fast the reaction happens in each. So to do the actual assay, we're going to take this serial dilution, and we're going to add 1 mil of starch to each of the tubes in rapid succession, as if we were able to do them all at once. And then we're going to wait 15 minutes for the reaction to happen, and then we're going to test with iodine. So we're going to find out which tubes the reaction has completed. Now, let's look at what we might predict. In the tubes on the left, like tube 1, we have more enzyme. We have more concentrated enzyme, and therefore we should expect to have a faster reaction. Because of the faster reaction, we should have less starch present at the end after 15 minutes, and therefore we should have a negative reaction with iodine. Okay? Over on the right, we have less enzyme, therefore we have a slower reaction rate, and we should have more starch present at the end. Okay? And since we have more starch at the end, we should have a positive reaction with iodine. Okay? So what you will find as you do this is that a number of tubes on the left have turned out kind of a pale yellow color, indicating no reaction with iodine. And then somewhere in the middle, we should hopefully somewhere in the middle we should start to see a reaction with iodine. And what we're looking for is the tube in which has the least concentrated amylase that still manages to complete the reaction. Okay, so we're looking for the first colorless tube, or the tube furthest to the right that is still colorless. Okay. Okay, in part 2 of this lab, we're going to basically do the same reaction, but instead of giving it a set time that we have the reaction going, we're going to monitor the reaction as it progresses through time. Okay, so the way this is going to work is you're going to have a spot plate. This is a plate with sort of dimples in it, and into each of those dimples you're going to add a little bit of iodine. Okay, so you're going to add iodine to each of these spots, and then we're going to start the reaction with a given pH. So let's say we're doing the reaction at a pH of 7. Okay, we're going to start the reaction, and then every 30 seconds we're going to take a little bit of the solution that's reacting. We're going to take it out and put it on this spot. Put it in the spot plate, and if the reaction is still happening, that is, if there's still starch present, then we expect the iodine in the spot plate to turn purple. Okay? So, and we're going to continue this every 30 seconds. We're going to take a little bit out every 30 seconds until the reaction is complete, until there's no more starch present, and these yellow spots, sort of pale yellow spots, they remain yellow. Okay, remember iodine is yellow. So, and then we're going to record the first spot in which there's no starch. Okay, so let's see what this looks like. And I want to say that we're going to do this in these little tiny microcentrifuge tubes, and we're going to do this in a hot water bath so that it happens. The reaction is happening at 37 degrees Celsius body temperature. So in this case, after we started the reaction, after all of the ingredients were in, we took some out and put it in this spot here. Okay, that's our 30-second marker. Okay, then we left this in the warm water, and after 60 seconds, or 30 more seconds, we took some out and put it here. Now, since this reaction is only starting, we expect there to be still some starch remaining, and so the starch will react with the iodine to form this purple blue-black color. After a minute and 30 seconds, there's still starch present. The enzyme is working, but there's still starch present, and so it's still blue-black. After 2 minutes, 2.5 minutes, it's still blue-black. But after 3 minutes, we notice that we don't get a reaction with the starch. Okay, that indicates to us that the enzyme has broken down all of the starch into maltose, and the reaction is basically complete. Okay, so we would record that as being complete within 3 minutes. Okay, and so we're going to, oops, not there yet. We're going to do this once for every single pH, and then we'll plot those on a graph. And we'll look for the optimal pH that the enzyme works in. Okay, in the third part of the lab, that we'll almost certainly, we'll do this next week. We're going to be looking at how the effects of the substrate concentration on the reaction speed. Okay, so we're going to, instead of, remember in part 2, we basically set up each reaction to have the same concentrations, and we just varied the pH at which the reaction was happening. In this third part, we're going to keep the pH the same, but we're going to vary the substrate. Okay, we're going to vary the substrate over a range, so that each time we do the assay, we can calculate a speed of that the reaction happens at. Okay, we'll calculate the speed that the reaction happens at, and we'll be able to plot this on the graph. So each assay that we do will represent one dot. Okay, and we'll hopefully get a graph that looks something like this, so that then we'll use R to fit this equation. Okay, to fit this equation to the data that we have here. Okay, and that will look something like this. Okay, so we'll fit that equation to fit that line to these data points and get something like that, and we'll be able to calculate all of these values here, include Vmax and the Km. Okay, and really what we're interested in for this lab is just the Vmax, the maximum rate at which the reaction happens, and we'll be able to estimate that using the data points on this line. So we'll find out whether they form this line or a line that's much more shallow, or maybe it rises really quickly and then flattens out. Fitting the line to our data will solve that problem, and so we'll be able to calculate Vmax.